DNA in the nucleus goes through a process called transcription, where the enzyme called helicase unzips the DNA strand and produces a template of matching DNA bases in a strand called mRNA. Matching the bases from the DNA strand to the mRNA strand is called transcription. Once the mRNA is made, it will leave the nucleus and enter the cytoplasm. Cell organelles called ribosomes floss the mRNA through using codons, which are 3 bases, to signal the tRNA to bring amino acids over to the ribosome. The building of the amino acid chain is called translation, and the chain that is formed is a polypeptide or a protein.
In DNA replication, the helicase unzips the DNA to create a leading and lagging strand. First, the primase comes and lays a small section called a primer. This marks the start of the new strand. Then the polymerase binds to the primer and makes the new strand, adding bases in the 5 to 3' direction. In the lagging strand, after the primers, the polymerase can only fill in small chunks called Okazaki fragments. After, the endonuclease removes all of the RNA primers in the lagging strand, and the polymerase fills them in with bases. Finally, the ligase comes and seals the DNA strands to form a continuous double strand. In the strawberry extraction lab, the soap and salt break open the lipid membrane, letting the DNA flow freely. The ethanol was used to bring the DNA to the surface of the solution.
The restriction enzyme depicted by the angry Pac-Man is a cutting enzyme known as endonuclease. It cuts part of the DNA in specific sequences and is cut staggered. When cut this way, a part of the strand overhangs to be able to connect with other similar strands as base pairs. That is the restriction site. The polymerase chain, or also known as PCR, creates multiple copies of a specific gene fragment after the section was cut out. Mostly used in forensics, the example on the board is also based on forensics and has 3 different copies of DNA, as one is from a crime scene and the other 2 are from suspects who may have been at the scene during that time. In a test tube, DNA strands are heated up to be able to be separated. Then, after they cool down, the primers begin to bind with the DNA, and the DNA is then heated up again for the polymerase to be able to complete the strand by extending the primer. This cycle is repeated multiple times where the DNA grows exponentially, to which the given result is the multiple copies of the genes.
This representation, the control represents where the dashes should be based on the number of base pairs. In person 1 and 2, the dashes are at 40, which means that they are closer to negative due to the higher amount of base pairs, whereas in person 3, the base pairs are 10 and it is closer to positive.